Here we go. And we're rolling.
Hey guys, this is Will, and I'm going to be explaining the cell cycle and mitosis and meiosis. So starting off with the cell cycle, the longest part of the cell cycle is going to be called interphase, and it will take up 90% of the cell cycle. And then we're going to break down interphase into three things. The first one is called gap 1, and this is where the cell is going to grow and develop. It's also going to assemble carbohydrates, lipids, and proteins. And then the last step in this phase is the mitochondria and the chloroplast are going to divide by binary fission. And then the second step in interphase is called the S phase, and this is where DNA is going to be replicated. A protein called histone is going to cause the DNA to coil, and when the histone and the DNA are together, this is called a nucleosome. And then the final step in interphase is called the gap 2, and this is where DNA replication is checked. So it's made sure that there's no mistakes. Everything is is finalized. And then in this step, the proteins that drive mitosis are going to be produced. And now we're going to do a brief overview of mitosis to start. And mitosis is also referred to as the nuclear division. And this occurs in somatic cells and is an asexual process, which means that it's only one parent, it's not going to be two parents. And then the final step of the cell cycle is a short one called cytokinesis, and this is the division of the cytoplasm. And then of course it's a cycle, so it's going to start over. So now we're going to go more in depth into this mitosis. The first step of mitosis is called prophase, and this is where the chromosomes are going to coil and become visible. And then the sister chromatids are going to join at the centromere. And then another thing that happens in this is microtubules form spindle fibers. And then the last part is the nuclear envelope is going to break into vesicles. The second step in mitosis is called metaphase. And this, actually, in between prophase and metaphase, you're going to have your prometaphase, which is going to orientate the sister chromatids to opposite poles. And then the chromosomes are going to line up at the equator. And those are going to be attached to your spindle fibers that were formed by microtubules. And then the next step is going to be anaphase. And in anaphase, the spindles are going to shorten so that the chromosomes, or sister chromatids, are separated on each part of the cell. And then kinetochores are going to disassemble the microtubules, thus disassembling the spindle. And then the sister chromatids are going to separate to their opposite poles. And those sister chromatids are now called chromosomes. Now, the last step is called telophase, and this is where the chromosomes are going to finally detach from the microtubules. The chromosomes are going to unwind and become thread-like chromatin. And then that nuclear envelope that broke into the vesicles is going to reform. Sorry about that. That's just Johnny.
Keep on going. Sorry.
And now in animal cells, the last step, cytokinesis, the cell is going to split by something called cleavage, which is where the cell is basically just going to have like a thing that just splits it. It's just going to split into two. And then in plants, you're going to have something formed called a cell plate that is going to divide the cell into two different cells. And then the final product of mitosis is going to be two diploid or 2N cells, which means there's double the chromosomes. Now another way of cell division is called meiosis. And this happens in germ cells. And this is going to produce genetic variation. Now in mitosis, they produce only clones, which means that the parents are just going to produce like miniature them. They're never going to change. But in meiosis, this genetic variation is going to cause differences in each generation that could help them survive. And it's going to help with it'll come to me. Sorry. Natural selection. They'll be, yeah. Alright. Now meiosis is very similar in how it works to mitosis, except it's going to start by doubling the number of chromosomes, and the goal of it is going to be finally to reduce the chromosome number by half. So you're going to have your same steps. You're going to have prophase, metaphase, anaphase, and telophase, and then cytokinesis. Except the first time they're going to be called prophase 1, metaphase 1, and so on. And in prophase 1 and metaphase 1, the only difference is that you're going to have twice the amount of chromosomes being split. So in this one, you're really, if it was a human, here you would actually have 92 chromosomes instead of 46. And then when you split down into your two cells, you're going to have 46 in each. And then once you get down to your prophase 2, metaphase 2, anaphase 2, etc., the same thing is going to happen, except you're going to go from 46 chromosomes down to 23 chromosomes in 4 different cells. So finally, you're going to go from 1 cell to 4 cells. And your 4 cells are all going to be haploid, or N, which means they're going to have half the number of chromosomes. Now, this is part of sexual reproduction, so these cells, when they're fertilized, will become diploid and thus have a full set of chromosomes. And I believe that is it. And I hope that's helpful.